Hello everyone, my name is Pavitra from Excelogen Biotech and I am here to present on the topic, that is to give a brief introduction on the topic next generation sequencing. In 1944, Oswald Theodore Avery proved that DNA is a genetic material. We all know that. In 1953, Watson and Crick model for DNA came, in which the DNA was described to be double helical strand structure. That led to the discovery of central dogma of molecular biology. Genomic DNA defines a species and individuals, which makes DNA sequence fundamental to the research on the structure and function of the cell and the decoding of the life mysteries. Next generation sequencing help biologists and healthcare providers in broad range. That is, their applications include molecular cloning, breeding, finding pathogenic genes, and comparative and evolutionary studies. And also, next generation sequencing have a broad range of advantages, which include they are fast, accurate, easy to operate, and cheap. NGS have developed tremendously in recent years, and it acts as the engine of the genome era, which is characterized by vast amount of genome data. In 1977, Frederick Sanger put forward a DNA sequencing technique that is based on chain termination method, that is with the use of dideoxynucleotides, and Walter Gilbert have used chemical modification of the DNA and subsequent cleavage at specific sites. Due to the high efficiency and low radioactivity, Sanger's was adopted as the first generation sequencing technique. At that time, DNA sequencing was laborious and radioactive materials were required for sequencing. After years of improvement, Applied Biosystem introduced first automated sequencing machine, that is AB370, in 1987, which used capillary electrophoresis, which makes sequencing faster and more accurate. AB370 can detect 96 bases one time, 500 kilobases a day, and the read length could reach up to 600 bases. Later, AB3730 XL was introduced, which can output 2.88 million bases per day with a read length of 900 bases. In 2001, we all know the Human Genome Project, automated sequencing instruments and associated software using capillary sequencing machines and Sanger sequencing technique. Human Genomic Project was established, which led to the powerful novel sequencing instrument to increase speed and accuracy, reducing cost and manpower. NGS makes genomic sequencing handy, but the followed data analysis and biological explanation are still bottleneck. Followed by Human Genome Project, 454 was launched in 2005. In 2006, that is the next year, Solexa was released by Genome Analyzer. Then sequencing by oligoligation detection, that is SOLID, from AgentCode. Later in 2006, AgentCode was purchased by Applied Biosystem and 454 was purchased by Roche. Solexa was purchased by Illumina. Here are few advantages and mechanisms of the sequencers. The sequencers include 454 GSFLX, HiSeq 2000, Solid V4, Sanger 3730 XL. And in this table, we can see sequencing mechanism, read length, accuracy, reads, output data, time, advantage and disadvantage. And that is a brief introduction about next generation sequencing. Thank you.